Hello everyone. Today we are going to learn about pour plate technique. Okay. So in our previous video we have already covered spread plate technique and streak plate technique. Okay. So if you haven't watched it, the link is in the description. You can go and check it out. Now in this video we'll focus on pour plate technique. So as we know that the types of microbial technique that we know today, there are three major techniques that are commonly known, right? So first one is the spread plate, second is the streak plate, and the third one is the pour plate. Now pour plate is one of the microbiological laboratory techniques that are used for isolating and counting the viable microorganisms that are present in the liquid sample. Okay, so here the liquid sample is very important. Without the liquid sample, the pour plate method cannot be executed. Okay, so in order to use the pour plate technique, you need to have your sample in the liquid or in suspension. Okay, now this method is very suitable for facultative anaerobic and microphilic microorganisms, right? So in the sample, if you take any sample, if you take, there may be different types of microorganisms. There may be aerobic, there may be anaerobic, microaerophilic, facultative, obligate. Okay, so different types of microorganisms may be present. Now this pour plate is very much suitable for growing all these types of microorganisms in vitro, right? So this is a very simple, less resource consuming, easy and very economical method. However, as I said, it requires the sample to be in liquid or suspension. So suppose you have a solid sample, if you have, then you need to make a liquid suspension of it. Okay. Now this method was also developed by Robert Koch, just like the other two methods. Okay, so all the types of microbial plating methods has been developed by Robert Koch in his laboratory along with other two scientists. Now coming to the principle of our pour plate technique. Okay, so the pour plate method is based on the fact that when an agar medium which is mixed with microorganism, if that is incubated, each of the viable microorganisms will multiply forming a separate colony. Okay. Now in this method, a certain volume, usually 1 ml of serially diluted liquid sample is mixed properly with approximately 15 ml of specific molten agar medium, which is cooled down to 40 to 45 degrees Celsius in a Petri plate. Okay. So this is the principle. Now this will become more clear if we look in our next procedure part. Okay. So basically here the agar medium is used where the liquid sample will be mixed with the agar medium and will be poured in the Petri plate, right. So coming to the requirements, what do we need? So first we need a culture media containing the agar, the sample of your interest. You prefer the liquid sample. If you have a solid sample, you need to make your suspension. Then you need a flask for making the media. You need a Bunsen burner for heat sterilization. Serially diluted liquid sample, the sample which you have, you need to do the serial dilution of it. You need micropipette, Petri plates, incubator, autoclave, distilled water and 70% ethanol. Okay, so these are the requirements that we need for this procedure. Okay, now there are two types of pour plate procedures. Okay, now both are easy. You can follow. You can follow either of them. Okay, so coming to the first procedure of this technique. So here in step 1, what you do, you prepare the required agar media first and autoclave it at 121 degrees Celsius 15 lbs for 15 to 20 minutes. Okay. Now after that you allow this media to cool down. In the conical flask only you allow it to cool down to 40 to 45 degrees Celsius, but you need to make sure that you do not let it solidify. Okay, you just cool it down to 40 to 45 degrees Celsius. Now after that, in a tube with about 15 ml of molten media at a suitable temperature, add 1 ml of sample. Now what you do, the sample, the liquid sample which you have done the serial dilution. Now from that serial dilution, suppose you want to take 10 to the power minus 4 serially diluted sample. Okay, so from there you take 1 ml of your liquid sample and you put it in this media which we have cooled down to 40 to 45 degrees Celsius. That should be approximately 15 ml, not more than that. Okay, 15 to 20 ml it is sufficient for the Petri plate. Okay. So, what you do, you take 1 ml from the serially diluted sample and you put it in the 15 ml of your media in the conical. Now, after that you mix the sample properly with the media by gently swirling. Now, after mixing, you pour the media into the sterile Petri plate. The plate you have autoclaved, the blank Petri plate which you have, what you do, you pour the media, the whole media, you pour it in the sterile Petri plate. And then after that, you close the lid of the Petri plate, allow the media to completely solidify. And after that, you incubate the plate in an inverted position at suitable incubation conditions. Okay, mostly for 24 hours at 37 degrees Celsius. Now, this will become more clear if we look into the pictorial flowchart. Now, here you have made the media. Okay, you have prepared the agar media around 15 ml and then you have done the autoclaving, and then after that you have cooled down the media to 45 degrees Celsius, right. Now, after cooling down, what you do, you have taken the serially diluted sample. You take 1 ml from this using a micropipette and you mix it in the media which you have cooled down to 45 degrees Celsius, right. So, you take 1 ml from it and you put it in the media which you have prepared. Now, after that you pour the complete media on the Petri plate. Okay. Now, you have mixed it properly in the conical and then you pour it in the Petri plate. Now, after that you close the lid and then what you do, you allow the media to completely solidify in the laminar itself, and after that you put it in the incubator in an inverted position for 24 hours at 37 degrees Celsius. So, this is the general temperature and time period which we follow, and you put it in the incubator and you do the observation the next day. Okay. So, this is the first procedure of the pour plate technique. Now, coming to the second procedure of the pour plate technique. So, here the most of the steps are same, half of the steps are same. First is the prepare the required agar media and autoclave it as usual, and then you allow the media to cool 45 to 45 degrees Celsius. Remember, you don't solidify it in the conical itself. Now, what you do, you dispense 1 ml of the diluted sample at the center of the Petri plate using sterile micropipette. Okay. So, here what you do, you take 1 ml of the serially diluted sample using a pipette and then you put it in the blank Petri plate at the center. Okay. Now, after that you pour the media in the Petri plate which you have cooled down to 45 degrees Celsius. You pour the media above this 1 ml which you have poured in the center of the plate. You pour the media over it, right, and then you close the lid of the plate and mix the sample properly with gently swirling of the plate. Now, this swirling should be done in the shape of S or H. Okay. So, you need to mix it very smoothly so that no bubbles form in the plate to avoid contamination. Okay. Now, after that you allow the media to completely solidify and then you incubate the plate in an inverted position at the incubation conditions as we have. Now, coming to the pictorial flowchart of this procedure. So, we have made the media first and then what we have done the autoclaving and then we have cooled it down to 45 degrees Celsius, right. Now, after it we have taken this serially diluted sample of the liquid sample which we have taken the serially diluted one. We take 1 ml from it and we pour it in the center of the blank Petri plate. Okay, you put this 1 ml of media at the center of this Petri plate. Now, after that you pour this cool down media which is cooled down to 45 degrees Celsius, not solidified. You pour the media over this one, over the 1 ml sample that you have put it at the center, right. So, you pour the media completely. Now, after that you close the lid of the sample gently, you close the lid of the plate over the sample and then you do the mixing. You need to do the swirling. Okay. So, in this manner you can do the swirling. Okay. See, you can see this is in H shape. So in this way you can do the swirling of the sample in the plate. Okay, and you make sure that you avoid any types of bubbles, right. So after mixing it down, you solidify it in the laminar itself, and then you incubate it in the incubator in an inverted position for the same time and same temperature. Okay, so this is the flow chart of the second procedure of the Petri plate. You can follow either of them, right. So now coming to the observation which we have in the next day, which we'll see. So this is how the plate was incubated. This is the pour plate before incubation. Now after incubation, we will see there are different types of microorganisms that have grown. Okay, so here different types of microbial colonies we can see with different colors. Okay, they are already dignified, shown as different colors, shown in different colors. So here you can see that some of the microbial colonies has grown on the surface. So these are the colonies that are grown on the surface. These are the aerobic bacteria. They generally need oxygen, therefore they grow on the surface of the media, right. Now here this type of colony we can see, this is half embedded in the media we can see. So this is the colony that are grown on the subsurface, or we can say these are half embedded in the media. These are the type of facultative, or we can say microaerophilic. Okay, these are the they need half of it needs oxygen and some of it doesn't. So these are type of facultative, or we can say microaerophilic types of microorganisms grow in this type of colony. Now here another colony we can see which is completely embedded in the media. These are the types of anaerobic bacteria. They don't need oxygen to grow. They are completely embedded in the media itself. Okay, so these are the different types of microbial colonies we can see if we use the pour plate technique. Now here you can see that the bacterial colonies grow on the surface, subsurface and embedded in the media. Okay, so these are the observation which we can expect after the pour plate technique. Now here are some of the precautions that we need to keep in mind. The first one is you need to autoclave the media, Petri plates, broth and distilled water, everything in the autoclave beforehand. Cool down the agar media to 40 to 45 degrees Celsius before mixing the liquid sample. Okay, don't mix your liquid sample at 60 degree or 70 degree, or after bringing it out from the autoclave. You don't mix your sample with it, because the microorganisms that are present in the sample might get destroyed or damaged or hampered. Okay, so you need to cool it down. Then you need to heat sterilize the top or mouth of the conical before pouring the media in the Petri plate. Right, when you remove the cotton plug, you need to sterilize the mouth of your conical and then you need to pour it on the Petri plate. Now add only 1 ml of liquid sample to the media. Now 1 ml of liquid sample is enough for the microorganisms to grow. Don't use more than that. Now mix the liquid sample with the media gently to avoid bubble. This is a very vital part in this pour plate method. You need to mix the sample very gently. The mixing, the more gentle and more proper the mixing is, the more space is there for the microorganisms to grow separately into the colonies, right. So you need to mix the sample first and then you need to mix it very gently as to avoid the bubbles, because bubbles can give contamination, right. And the last one is allow the media to solidify before incubation. Suppose you have put the media on the Petri plate, you have mixed it properly and then you have put it immediately in the incubator. This is not a proper process. Okay, you need to solidify it after closing the lid in the laminar itself, and then you put it in the incubator. Okay, so these are some of the precautions that you need to keep in mind in the pour plate technique. And that's all for today's video. I hope you have understood today's video very well. If you have any queries regarding this video, don't hesitate to put a comment down below. And if you need the details of this video, notes of this video, you can follow our LinkedIn page. The link is in the description as well. And if you have liked this video, give it a thumbs up and do share and subscribe to our channel for more such microbiological updates. I'll see you in a next video.